welcome to my channel, that is Applicons of Biotech. Firstly, thank you so much to all students, young researchers, scientists, teachers and my all friends. I'm so glad that you liked our videos and thank you for your support. Okay, today's topic is very important for those students who are preparing for entrance exams in science field. Okay, let's move on to our today's topic. So, everybody knows that gene silencing is possible, but some of you are not familiar with how we can silence or why we silence gene and what is the purpose. All of your questions answers are in this video. First, I will explain you why we silence gene and their purpose by giving one example. Our example is of mosquitoes. These research could be the birth control methods and they need to control mosquito population. So, for this, the University of Arizona have discovered a protein in mosquito that is important to the process of producing viable eggs. So, the question is why we need to control mosquito population. Worldwide, more than 500 millions of people suffer from diseases transmitted by mosquito like malaria, dengue fever, Zika, so many, and nearly a millions of deaths are attributed to mosquito-borne illness each year. What they did, they silenced the gene that is EOF1, the eggshell organizing factor 1 in female mosquito. These genes are silenced by RNA interference. The mosquito laid eggs with the defective egg shells and leading to the death of the embryo inside. That means they are successful in silencing those genes. Let's move on to the next slide. It wasn't a direct discovery. The RNAi was accidentally found in plant, that is Plutola hybrida. Some researchers tried to obtain transgenic proteinuas with a greater amount of anthocyanin pigment by amplifying the gene activity of cyclone synthase. And instead of obtaining deeper purple color in petals, the white or chimeric flowers were produced. As you can see, this is the result of their experiment and this was a goal. The next one, the definition. The definition of RNAi is a process which targets the mRNA strand and degrade them with the help of double-stranded RNA, and it is found in plant, that is fungi, and also in animals. In different organisms, it named by different term. In plant, it is called as post-transcriptional gene silencing, and in fungi, it is called as quilling, and in animal, it is called as RNAi. Now, it is time to know about discoveries. The first one, in 1999, the co-suppression phenomena in Petunia hybrida was found. The second one is similar phenomena were named as quilling in fungi, was found in 1992. And in 1998, in C. elegans, they were found that the presence of double-stranded RNA. That means the RNAi phenomena. And finally, in 2006, the Craig Milo and Andrew Fire got the Nobel Prize. The next, it is the component in RNA mechanism. The first one is the RIX complex. It is an RNA-induced silencing complex. The second one is double-stranded RNA or siRNA or miRNA. And the third one is dicer. The dicer is RNA's 3 type of enzyme. What it means? They are specific to double-stranded RNA. And fourth one is slicer and argonate. The slicer is RNA's H type of enzyme. The argonaute is of RNA's 3 type of enzyme. And these 2 enzymes work in association, and then their work will be seen on next slide. Inside the cell, if there is a double-stranded RNA produced, then the RNA process is possible and the breakdown of protein coding is possible. If there is a production of double-stranded RNA, then this dicer enzyme plays a major role here. As a dicer is RNA's three type of enzyme, that is endonuclease enzyme specific double-stranded RNA. And that means this specifically binds to the double-stranded RNA and leads to the production of siRNA, miRNA and shRNA. Next, in this image, you can see this RISC complex is produced after producing this siRNA. And this dicer and they bring out the gene silencing process by combining many other proteins together, and that protein complex called as RISC complex. Next, in this diagram you will get clear idea. First, the dicer is cut the double-stranded RNA into the siRNA, and then siRNA and some of the proteins produce a RISC complex, and this RISC complex unwind the siRNA, and this siRNA get attached to the RISC complex. The major task of dicer is to first produce siRNA, miRNA and shRNA. And most of the siRNA and miRNA are approximately 21 nucleotides long. Once we produce RISC complex, this siRNA then binds an organoid protein and degrade the miRNA. And the one strand of the RNA is selected and remains bound to the organoid. This is called guide strand. And the combination of siRNA and organoid along with the slicer and the other proteins is called as RISC complex. And this siRNA directs the RISC complex to bind to the specific messenger RNA. This targeting is very precise because it determines by base pairing. But between the siRNA and target mRNA, the siRNA often have perfect complementary to their targets. And when it's finally bound, the organoid catalyzes the cleavage of messenger RNA, which can ultimately degrade the mRNA. That means no RNA produced and no mRNA. Next, now I am going to tell you about my master's research work. It was on RNAi, and I will not explain you in detail, but I will explain you some concept in short. So, my research work was on Plutola xylostella. It is an insect pest. It infests the cruciferous crop, and it is most destructive crops in such as cabbage, broccoli and cauliflower. These pests destroy the whole field by eating the cabbage leaves, and it is creating resistance against all chemical pesticides day by day. So, I targeted 2 genes to silence based on its normal physiological importance for its development. And the genes are the tyrosine hydroxylase and EGR, and I got very promising result. The next is application of RNAi. The first application is abiotic stress tolerance. In abiotic stress tolerance, let's take one example of drought stress. The idea here is to silence those genes responsible for affecting the plant health. See, if plant identifies a stress, the signaling pathways are going to be activated, and some transcription factor and proteins are responsible for wilting, like in drought condition. So, to stop wilting, we use RNA interference and silence those genes. The second one is prolongation of shelf life. It was done in tomato. To increase the shelf life of tomato, they use the RNA interference. The variety known as flavor saver tomato. The third one is modulation of flower color and scent. To change the color of flower and to increase the scent, they also use the RNA interference. It was done in Tornia hybrida, Gerbera hybrid, Gentiana triflora. And next is seedless fruit development. It was also done in tomato. The next is biotic stress tolerance. The biotic stress tolerance, that is for crop protection and the pest management purpose. And the last one is nutritional development. To increase the nutrient in food or in plants, they use RNA interference. That is to increase the vitamins, zinc, iron or carotenoid contents. That's it. This is the end of RNA interference topic. Thank you for watching video. I hope you understood this concept. If you understood this concept, please hit the subscribe button, like this video and press the notification bell so you will get more videos. And you can follow us on Instagram page, that is Amplicons Biotech. That's it.